Good morning and welcome to the EB-101 Clinical Development Update and Discussion. I'm Jen Dawson, Pedra's Outreach Manager. I will be moderating a Q&A at the end of this session, so please submit your questions via the Q&A button on your Zoom screen. A recording of this webinar will be posted on Pedra's website following today's presentation. As we begin, I'd like to start by thanking Abeona Therapeutics for sponsoring this webinar and for providing speakers and content. I am pleased to introduce our speakers today. First, I'd like to introduce Michael Amoroso. He's the Chief Commercial Officer at Abeona. Mr. Amoroso joined Abeona as Senior Vice President and Chief Commercial Officer in July 2020, bringing over 20 years of product commercialization experience in the biotechnology and pharmaceutical industries with a focus on cell and gene therapies and a proven track record of launching innovative drugs for rare diseases. Prior to joining Abeona, Mr. Amoroso held various senior-level commercial positions at leading biopharmaceutical companies, including Kite, Eisai Inc., Celgene Corporation, now a subsidiary of Bristol Myers Squibb Company, and Sanofi. Mr. Amoroso earned his MBA in Management from the Stern School of Business, New York University, and his BA in Biological Sciences, summa cum laude, from Rider University. I'd also like to introduce Dr. Jean Tang. She is the Professor of Dermatology at Stanford University Medical Center and Principal Investigator for EB-101 Pivotal Phase 3 Vital Study. Dr. Tang's research focuses on genetic skin diseases such as basal cell nevus syndrome and epidermal mollitus bullosa. She studies new ways to treat and prevent NSMC and melanoma. Dr. Tang has led or co-led the conduct and completion of 6 investigator-initiated clinical trials in BCC and EB. She received her MD PhD from Stanford in biophysics, completed her dermatology residency at Stanford, and then went on to UCSF for a 3-year postdoc in mouse genetics, while simultaneously pursuing formal coursework in biostatistics, epidemiology, and clinical trial design in the KL2 CTSI program. Thank you both so much for joining us today. With that, I'd like to turn it over to Mr. Amoroso.
Good morning to all healthcare practitioners. It's a pleasure to be with you. Thank you for the very wonderful introduction, and I hope you're all enjoying and coming off the back of a successful Pedra annual meeting. My name is Michael Amoroso. I'll be taking you through some details about Abeona this morning. Today's agenda, we'll talk a little bit about Abeona and EB-101. I will then hand off to our world-renowned physician, Dr. Jean Tang, our PI out of Stanford, and there'll be a question and answer portion of the program. Today, during the course of the presentation, I will provide some content that will have forward-looking statements pursuant to the safe harbor provisions of the Private Security Litigation Reform Act of 1995. These statements are based on assumptions that we believe are reasonable, but such forward-looking statements may prove to be incorrect. About Abeona, our mission, to work together to deliver and unlock the promise of gene and cell therapies for patients afflicted with serious disease. The strategy is to harness the promise of genetic medicine to transform the lives of patients like those we'll talk about today with recessive dystrophic epidermolysis bullosa. Our mission is simple: one compass, to realize a world where cure is the only acceptable new standard. A bit about Abeona. Abeona is a fully integrated gene and cell therapy company. We have comprehensive gene and cell therapy capability, a late-stage program in our phase 3 VITAL trial, which Jean will take you through today, in our study for EB-101 in patients with recessive dystrophic epidermolysis bullosa. We have breakthrough therapy with gene-corrected cell therapies. We also have our very promising AAV 9, and proprietary AIM programs in gene therapy, our lead programs in phase 1/2 clinical trials for MPS IIIA, IIIB, otherwise known as Sanfilippo disease. Our gene and cell therapy expertise in manufacturing out of Cleveland, we believe, is world-class, and we have a robust pipeline and clinical stage as well as preclinical programs with our AIM AAV capsid platforms. Today we'll focus on our lead program in EB-101 in recessive dystrophic epidermolysis bullosa. This program, we've met with regulators around the world. The FDA, we have the Regenerative Medicine Advanced Therapy designation, which allows us to communicate with the FDA, as well as breakthrough therapy early and often to try to bring solutions to patients faster, as well as we have EMA orphan designation, as well as that with the FDA. A bit about EB-101, an autologous gene-corrected cell therapy product. In RDEB, we know the defect comes from the lack of the collagen 7A1 gene, unable to produce type 7 collagen, the anchor for the dermis and epidermal layers of the skin. At Abeona, what we do with EB-101 is we take biopsies from each patient of the non-afflicted surface area. The patient's skin cells are then expanded in vitro in our facilities in Cleveland and transduced with a functional type 7 collagen gene, correcting for the disorder, to produce EB-101 sheets. These cells grow over a series of almost 16 to 20 days. EB-101 sheets are then returned to the clinic after about a 28-day interval to the very capable group at Stanford, where they're transplanted onto the wound sites, these new gene-corrected therapies. Our typical yield is about 6 sheets per patient, each about 40 centimeters squared. Think about a typical business card, a little bit bigger, times 6. So we're able to treat up to 240 centimeters squared of one EB-101 administration. And the production of type 7 collagen followed by EB-101 transplant can lead to long, durable wound healing and an improvement in morbidity such as pain and itch. I'm very excited now to turn the presentation over to Jean, where she could tell you more about our pivotal VITAL trial that's ongoing. Jean, please take it away.
Hi, everyone. Hi, everyone. Thanks, Michael, for that introduction. And I wish we could be together in person. I have lots of great memories of gathering at PEDRA in the scientific symposiums, the small breakout sessions, and of course the dinners and the food and the people. So I really look forward to seeing all of you guys hopefully in person sometime soon. But for the time being, I really appreciate you coming on to this webinar. I'll be talking about some of the phase 1/2 results for EB-101 and also about the phase 3. I don't need to tell this audience about the full burden of RDEB. We certainly know about the large chronic wounds, and sometimes these are the most debilitating, painful, hard to heal, often infected wounds of the patients. And as the patients age, they get more of these chronic wounds. They're at risk for squamous cell development. That is the clinical burden, but many of us who've taken care of RDEB patients also realize the huge economic burden. Even current wound dressings cost a patient and their family $245,000 or more yearly. And there's no question that in all of our quality of life studies and surveys that the humanistic burden of RDEB is huge on the patient and on the caregiver and family. So currently there are no approved treatments for RDEB wounds. There's a number of phase 3 clinical trials ongoing, which is just incredible and exciting. The purpose of EB-101 is really to address the underlying cause of RDEB, really replacing the collagen 7 where it's needed in the skin and to heal these chronic wounds. From our preliminary data from the phase 1/2, once we heal these chronic wounds, there's a reduction in pain, there's a reduction in itch, and hopefully a reduction in the reliance of opioids and other medications associated with these chronic large wounds. So oftentimes, the chronic wounds that we have studied average about 118 centimeters squared. So these are large wounds that are the most difficult to treat. Large wounds oftentimes on the shoulder, the back, and extremities. Many pediatricians and dermatologists might ask, why don't we treat recurrent wounds? We know these are smaller, these heal, they open up again and heal, but we really wanted to address and treat the wounds that were most bothersome to the patient. And in the phase 1/2 trial, we really optimized for what the patients needed. And even if the grafts were hard to take on the back, that's where we put them because that's the most problem area for many of the patients. So very briefly, EB-101, how does it work? In the past, I've spoken about this in prior PEDRA meetings, but I just wanted to give a quick overview. We take 2 small biopsies from non-wounded skin from RDEB patients. These biopsies are then sent to the manufacturing site of Abeona in Cleveland, where the keratinocytes are expanded, and then we use a retrovirus containing collagen 7 gene to infect the cells, and the schematic shows green cells going from gene-corrected purple cells. And this process to assemble 3D sheets and place them on a Vaseline gauze, the whole manufacturing process takes about 28 days. Once they're ready, we notify the patient. The patient comes in for general anesthesia, and a plastic surgeon then grafts on these 6 EB-101 sheets. The phase 1/2 study has been completed. We treated 7 adult RDEB subjects, all of them with severe disease. In these 7 subjects, 6 wounds were treated. So we've evaluated 42 wounds for an average of 4 to 6 years. So I want to speak first about efficacy and secondly about safety. In terms of efficacy, here is an example. Here's a large chronic wound that has not healed for 10 to 15 years. This patient complains of consistent pain, itch, and this is the most significant problem area for wound dressing. We took a small biopsy, created 6 grafts, and 9 months later, you can see significant wound healing on her upper arms. These are 2 grafts labeled C and labeled D. The other grafts were placed on the contralateral arm. In her antecubital fossa, we marked an untreated, ungrafted control wound, and you can see 9 months later, the untreated wound is still there. We followed this patient yearly, and on average, we see healing lasting for 3 to 5 years after treatment. Most importantly, this healing was associated with a significant reduction in pain and itch. Secondly, I wanted to talk about safety. We did not detect any SCCs on the graft sites. We did not detect any systemic immune reaction from these autologous grafts. And we continue to follow up the patients, and the patients report that they still have reduction of pain and itch, and they would like to receive more grafts. When we do serial biopsies during follow-up, we can detect using IEM or DIF continuous collagen 7 expression 2 years after treatment in some of the grafts. This is another set of photos showing a chronic wound on the hand at baseline. The DIF shows lack of the green line, which is an antibody detecting the NC2 portion of collagen 7. A biopsy taken from the graft site at 3 months shows nice wound healing and concomitant restoration of collagen 7, as you can see in the green line on the DIF specimen. At 6 months, you see nice wound healing. Also this green line showing collagen 7, and this is also detected at 12 months. This is a table summarizing the 6 wound sites that were treated in one patient. The location is described here. These wounds were present for at least 3 to 5 years, and the green box signifies that when we visually inspect the wounds and photograph them, that we see 75% or greater, often 100% wound healing consistently over time. The yellow box shows less than 50% wound healing, and this site happened to be infected at this time point, and then there was subsequent wound healing. The biopsies I showed DIF expression of collagen 7, but also we saw anchoring fibrils as well. I won't show that data here. Over time, we asked the patients, what is the level of pain at the graft site? And so in summary, about 53% of the graft sites, the patients reported pain, and you can see a significant reduction that's durable, a reduction in pain over time in these graft sites. This has been published in JCI Insight, but as a summary, what we are speaking about here is how much wound healing does it take to really make a meaningful difference for patients. From our phase 1-2 data, we realized that 50% wound healing was associated with quality of life improvements of pain and itch. So this graph shows a comparison between the treated wounds in the dark blue and the untreated wounds in light blue. And how do we measure success? You can measure success by wounds achieving greater than 50% healing over time, or wounds that achieve greater than 75% healing in panel B. The overall conclusion is that the treated wounds are much better than untreated wounds when it comes to 50% healing over time. Look at this result at 24 months. You still see much higher wound healing in the treated sites versus untreated. And if you use your marker of success as 75% wound healing, again, after 24 months or 2 years, the treated wounds are still do much better than the untreated wounds. I want to emphasize that the average wound area was greater than 130 centimeters squared at 3 and 6 months. So again, these are large areas and not small recurrent wounds. So what about the phase 3 study? Well, we're really excited about it. Stanford is one of the sites, but this is supposed to be a multi-site trial. So many of you pediatric dermatologists out there listening to this webinar, please do contact us if you're interested in being a site, and we'll do our best to set you up fast. This phase 3 trial intends to enroll 10 to 15 RDEB patients, treating approximately 30 chronic wounds in total. I'll go over the study design and the follow-up period. The study endpoints really focuses on the proportion of wounds with 50% or greater wound healing at 3 months. We'll also look at 75%. We'll also look at pain and itch during follow-up, similar to the phase 1-2. The key thing is patients need a clinical diagnosis of RDEB. They have to be 6 years or older, and they must have 2 wounds that are chronic. They have to be larger than 20 centimeters squared and present for 6 months. Why at least 2 wounds? Because as part of the discussions with the FDA as a phase 3 trial, we need to randomize paired wounds. So if you can imagine 1 wound here, 1 wound here, then we send a randomization scheme, and the randomization scheme tells us which site to treat versus which site to follow as control. So within one subject, wound pairs will be treated or untreated, and that's how the FDA will rate wound healing within a patient. Here is the schedule of events. The patient comes in around day 26 for 2 small biopsies. Then they have the option to fly home or stay here. In between, a home nurse visit will occur just to make sure the wounds are still present and not infected. When the biopsies grow into 6 grafts, then the patient's ready for grafting. They can fly back to Stanford, and at day 1, we assess and make sure that they are indeed still good surgical candidates. And then our plastic surgeon grafts up to 6 grafts to these wounds. The patient stays at Stanford inpatient for 7 days. There's 24-hour nursing care, not because this is a serious treatment, but we want to do everything we can to help the patient and the family protect the grafted sites, make sure the patient is comfortable, make sure the grafted sites remain uninfected. For the follow-up period, the patient can go home. There will be a number of phone calls and home nurse visits, and the patient comes back at 6, 12, and 24 weeks for wound evaluation to understand if the grafts really do contribute to wound healing compared to controls. We ask them about pain and itch, and we make sure that their labs continue to show things are safe. We are in discussions with the FDA, and because of COVID, there's likely a reduction in some of these study visits. The important thing to talk about your patients, if you have any, and please refer them if you do, is there's no cost to participate in the study. Abeona has graciously provided lodging, meals, transportation. The patient can stay at Stanford for, in between biopsy and grafting. Usually Abeona is able to find first class or business class seats just because of COVID to reduce the density of people on the flight. And of course, any caregiver or parent will travel, and meals and lodging will also be paid too. The Stanford EB team will be present at all the visits. We will help with wrapping, bandaging care. And like I said, we will take all the precautions to make sure that the patient and the family is safe. We at Stanford are tested weekly for COVID. All of our clinic visits are at reduced density just to make sure that social distancing happens. And really, whatever additional requests from the family and the patient that's necessary to participate, just email and call us, and we can probably accommodate. So, to the pediatric dermatologists out there, if you are a site that would love to participate, we would love to hear from you. If you have patients with RDEB and wounds, please email us, give us a call, and thank you for all of your help and your time. And we can't wait to heal these wounds in RDEB. These are, we know, some of the most deserving patients who've suffered a long time. And I'm just really excited that hopefully this year or next, we have something available for these patients. Thank you.
Hi, everyone. Can you hear me?
Yes, Jean.
All right.
Hi,
Yay.
Jean.
Hi.
Hi. Thank you all so much for those lovely presentations. So I want to transition over to our Q&A now. We're getting a lot of great questions. So I'd like to start with one of our investigators is asking, let's get back up here.
We have a number of Canadian dermatologists on there.
We do, yeah. So
in Canada, that's great.
Yeah, so let's start with that. Is this available and open to Canadian patients?
You want to talk about the pivotal, and then I can talk about the pathway to commercialization?
Oh, sure. So basically, if the patient is willing to travel to Stanford or other US sites, and as long as they have some form of traveler's insurance, then absolutely they can come and get treated here.
And then for availability, assuming a positive phase 3 pivotal trial, of course, we're starting in the US with the FDA, but we will be looking at expansion to get to as many of the afflicted as possible. That would be Health Canada, that would be the EMA, and maybe even Japan with the PMDA. Thank you.
Excellent. So I want to go to another question here. Is there a limit to the number and size of sheets that can be created from an initial biopsy? Would additional biopsies be needed should someone receive additional treatment in the future?
Sure. Gene, I'll let you speak to how many sheets the protocol speaks to today, the up to 6. And then what I'll do is I'll talk about commercialization again. We'll do it that way if that's okay.
Sure. If you recall, there are 2 biopsies taken, and the size of these biopsies are 8 millimeters. Just these 2 little biopsies are sufficient to make 6 sheets, and these 6 sheets are applied to wherever the worst wounds are. That is the purpose, and it's very well outlined for the phase 3 trial. But maybe this question speaks about the future. So there's no limit to the number of biopsies that one can take and then generate more sheets. So for example, if everything goes well, the patient has additional wounds that need to be treated, then we take 2 more biopsies and create 6 additional sheets. So this patient would have 12 sheets so far. But for the phase 3, it's 1 round.
Yeah, and I'll add to Gene's Gene's base and foundation. I think it's a great question. And I think our view collectively is that we need to treat as much of the body surface area afflicted of the worst wounds. Large wounds greater than 20 centimeters squared, chronic wounds open longer than 6 months. We know these are the wounds that take years and sometimes beyond years. They do not close themselves, lead to infection and sepsis, malnutrition, and ultimately squamous cell carcinoma. And most of these patients hit their demise between 30 and 40 years of age. So I think some of the follow-ons, EB-101, as Gene talked about through VITAL, will be 6 almost business card sheets for a total of they're 40 centimeters squared each, for a total of 240 centimeters squared. That is one product. If you look at the literature, from what we've seen from Stanford and other places collectively, and I know it's tough, we think the median patient, and Gene, correct me anytime, afflicted body surface area of RDEB is about 30%, some more, some less. So if you think about a product, we at Abeona, how we're thinking about it with Gene and team, one product of the 6 sheets would cover in the median patient about 10% of the problem. If you're on the lesser side of the median, obviously you can create more, and then vice versa if you're on the other end. So I think one of the things we're thinking about, remember, these are live products. They have a stability period of 36 hours today. We'll try to optimize that over time. We have to get them, the cultivated keratinocyte sheets, grown, sent to the surgical transplantation site at Stanford right now. So today, there's variability on how those cells grow between patients, very similar to CAR T and other cell therapy products. And we are, the initial product we believe will be up to 6 sheets. Whether we can optimize more of that space in our process development over time with stability, more time the product's viable, are things we'll always be seeking. And we have a retreatment protocol, phase 4 work that we're starting to get ready to roll out with Gene and team, which is 6 months after treatment. Not the same wounds, because the beauty of the product right now is we've had great durability. We'll see about retreating wounds if we have to in the long term. The phase 1/2A data showed wounds closed up to 5 years, 2, 3, and 4, 5 years. But retreatment being other areas that have not yet been addressed in the body is a phase 4 protocol. But want to just make sure I'm clear. To get an FDA approval pivotal, it is the 2 biopsies, it is 6 sheets today, which has a variability of how much of the afflicted surface area per patient it covers. Gene, please feel free to correct me if I'm missing anything.
No, that was really good.
Thank you. So can you tell us a little bit more about the cost of the product?
Yeah, the commercial guy in the company always get that question, and it's a fair question. And what I will share with you is the following. Look, folks, we don't know the cost. We don't know the cost until we have a final target product profile, which is what the phase 3 data shows from an efficacy and safety standpoint. We're incredibly hopeful from what we've seen from the phase 1/2. Again, a safe product with really durable wound closure. We know that adds to an improvement in morbidity, pain, itch. We want to prove that in VITAL in a pivotal setting. And then ultimately, the big question is how much of these most problematic wounds do we need to close for how long in order to change the prognosis, dare I say, of this disease? And that's the ultimate goal. So we don't have a price today. We have to see what the final product looks like. I'll share with you that just in wound dressing coverages, ointments, the hours, and the actual supplies, an average family between $200,000 and $250,000 today in wound closure. So this is financially toxic to families to this disease.
Mhm.
And I will just share with you the philosophy at Abeona. We believe in access to human beings. Get it to as many of the afflicted as possible. We do believe, of course, in the cycle of innovation and R&D and to pay for innovation. And we will try to be as value-based in our pricing. How much better are you than the standard of care today? And with the thought process of how do we get to as many humans afflicted? But I won't speculate on numbers and price because we genuinely don't know till we have a final target profile from our phase 3 data. I hope that gives you some clarity of at least how we think about going at pricing.
I think so. I appreciate that process. Can you remind viewers what the eligible ages are for participation?
I believe it's age 6 and older, and the minimum is 2 wounds of 20 centimeters squared size or greater. And the idea is we want to have 2 wounds, so one can be randomized to treatment with graft, and the other one is an untreated control. So age 6, we're really good at biopsies, at lab draws, doing whatever we can to try to make the pediatric patient and their family comfortable.
Excellent. What other sites are participating in the US?
So I'm going to take this one and roll it over to Jean, and I'm going to be shameless to the healthcare practitioner community. Folks, we believe deeply in the science. This program at Stanford with some amazing scientists there. The phase 1/2A data that we showed the long-term follow-up in July at the Society of Pediatric Derm. We believe it's incredibly exciting science, but without you, we can't make this happen in large scale for these patients. We estimate about 2,500 patients in the United States with RDEB today. Our only site right now is at Stanford. We believe we can accrue the pivotal fully at Stanford, although obviously that takes a little bit longer. We want the solution. We want to prove the efficacy and safety and then bring the solution to patients as fast as possible. But I've started to have a lot of conversations with the KOLs out there, the folks yourselves in the community. And I've asked you to think about if the product was approved tomorrow, knowing there are no options for children. I know there's some topical gels that potentially are coming to market. We are very pro life science company bringing innovation. There is no competition. We need a continuum of care, but we also need you to invest. We need you to invest of what would it take to be a commercial site. We know there's an OR procedure. We know there's a short stay in the hospital. So the Abeona team will be working very closely with you to make sure we can talk about what it takes to be a commercial site and potentially a clinical trial site. We had planned on opening a second site on the East Coast for practicality of travel for patients. That became difficult in the heart of the COVID-19. That was New York City. That became tough with the COVID-19 pandemic, but we do want to be opportunistic about onboarding next sites. I know Jean's passionate about that and getting the experience out there. So my message to you is please help us bring this to patients as fast as possible.
Yeah, I think what Michael said is right. I think there's a lot of excitement and engagement from patients and their families, but the challenge here is traveling to California. And as you know, many of our patients are maybe in Canada, maybe on the East Coast. And so whatever site is interested in starting this clinical trial, we would love to have you. Our plastic surgeon can talk to your plastic surgeons, anesthesiologists can speak to each other. All the protocol is written. We'll, give you the nuts and bolts and help with training, whatever it takes to get these patients treated in an easy and safe fashion.
Thank you. And I just want to let everybody know, if you are interested in becoming a site, feel free to contact Pedra or myself, and we will make sure that you get in touch with Mr. Amoroso and Dr. Tang. So let me know. A couple of other questions. How are patients and parents responding to this potential new therapy?
Jane, I'll let you speak to maybe the interaction, the anecdotal interaction. And I don't know if, Jen, you can unmute Jody, but Jody's in touch with families and the groups all the time. She can add some color also.
Absolutely.
Yeah, I think for the phase 3, we've treated, 3 patients so far. And as long as the sheets are protected well and not infected, I can tell you preliminarily, the wounds are healing. The patients seem to have a reduction in pain and itch on those sites. So the patients are quite happy about it. It does take time and effort to travel over here, right? So it's not necessarily easy for everybody, but our patients seem to tell us that it's been worth it so far, especially now with COVID, with remote school and remote work. You know, they've made it possible to travel to California. So like I said, whatever the family needs and accommodations, if they need to stay in a hotel for 4 weeks between the time of the biopsy and the surgery, we can figure that out.
Can you hear me?
Yes, Jody.
So I'm Jody Gillen. I'm the Vice President Patient Advocacy at Abeona, and I could just add a lot of patients who completed the phase 1/2 trial have reached out to me. They're very interested in retreatment, so that's always a good sign that they had a positive experience, a lot of interest in treating other wounds. There's also a great deal of interest in the trial. I know COVID put a bit of a wrench into things, a lot of concerns around travel that we're doing our best to mitigate. I think Jean mentioned a lot of those mitigation factors, whether they're, you know, first class seats on planes, making sure rows are empty, making sure that, you know, there's a private driver at Stanford, doing anything we can that's very much on an individual level, working directly with the patient and the family to understand what would make them the most comfortable, that would enable them to be part of the study. But the interest is there.
Excellent. Thank you. So I think one last question. Looks like we've got one more coming in. How many patients were in the phase 1/2 trial?
Yeah, we had 7 patients. Each patient was treated with 6 sheets. So we have evaluations on 42 wounds that were treated over close to 4 to 5 years. So again, phase 1/2, 7 adult RDEB patients, all of them were severe. The youngest was 18 with good safety and long-term follow-up data. And this is why we are allowed to start the phase 3 by the FDA.
Excellent. So I want to talk a little bit about what this means for early career pediatric dermatologists and expectations, and what they might, you know, what this means for EB in 5 to 10 years for our early derms.
Well, if I may take that,
Please.
you know, I've been in dermatology for 15 years now, and I can count on my hands the number of exciting technologies and revolutionary treatments coming out. And for us who know EB, we know the suffering, we know the clinical need, but if we switch onto our other hat, you know, people used to make fun of dermatologists as lotions and potions folks, right? What else can you do? But, you know, this is genetic personalized medicine at its best, right? We know collagen 7 is missing. You have a genetically elegant way of replacing collagen 7. The skin grafts are not a big deal by plastic surgeons. They use it all the time. It's not fancy technology. They use it on burn patients. So now you're marrying gene therapy technology in a safe way with skin grafts with patients with terrible wounds, right? So for me, this is in some ways low-hanging fruit. The science is proven, the methods are there. Let's just execute and get it done for our patients. And hopefully we have a sound body of evidence to talk to the FDA about.
Well said, Jean.
So if we could go back to the patient perspective a little bit, have you been consulting with or working with any of the patient advocacy organizations that support EB?
Jody, take it away.
Very, very closely. I think I'm in touch with them almost on a daily basis. So we actually also hosted webinars through Deborah, through the EB Connect platform and through EBRP. They're live on those sites. We're actively engaged, communicate regularly. Everything we do is hand in hand with our patient advocacy organizations, and then even Deborah International.
And one thing that's really important to Pedra and how we work across all the different groups with industry, advocacy, and our clinicians and investigators, what was it? Tell me a little bit more about how the relationship began and was established with Abeona and those advocacy organizations.
So I joined the company a year and a half ago. I know those relationships existed long before my time at the inception. And, Jean might even want to comment on the history of this product because it came from a partnership with an advocacy organization. But coming on board, those relationships were deeply entrenched in the company. Everything we do is in tandem, whether it's something that's visible, such as a webinar, but even things like regulatory meetings and even involvement in the PFDD of epidermolysis bullosa back several years ago. So, it goes both ways. The advocacy organizations are involved and partnering, with us in development, but they're involving us as a partner in things that they're doing as well. So we're in support of that, whether it's a gala or venture into cures, but also deeper projects such as a value project to demonstrate the burden of the disease. So it's a very deep relationship that goes in both directions.
I think the foundations are just so excited about any potential therapy reaching clinical trials, and they want to be supported. They've been generous with their time, and like Jody said, we've talked about the results using, the community webinars and email dispatches. When there's no therapy for EB, the foundations are very passionate about supporting, the advancement of any program almost. Jen, you had asked a question about young investigators in pediatric dermatology. I just wanted to follow up on that. This is a great trial to get involved in. Please join us as a site. Even if you are a young investigator, we will help you pair up with other senior investigators, or your institution's plastic surgeons and anesthesiologists. This is not rocket science at all, and this would be a wonderful opportunity to help launch your career. So please contact us.
Jen, the only thing I'll add to that, the only thing I'm going to add to that is, again, just for our HCPs, first of all, thank you so much for being with us. Second, in this ultra rare space that you all know better than I, it's going to take many hands. It can't be done alone. I think Jean Tang is amazing. I think what Stanford's done, the inception of EB, we couldn't be more appreciative about. But again, think about if there was an approval tomorrow for these patients, 2,500. If Stanford could do 20, 50, or 100 a year, we still can't get to those in need. We need your help. We need you to enlist. So that's no better time than starting in the clinical pivotal trials, but also maybe to think about what a world might look like with EB-101 in there for your patients. Thank you.
Thank you. I have one last question I want to go back to. If the audience has any more questions, please feel free to submit them. We're going to wrap up in just a few minutes here. But Jean, going back to the early investigators and how to become part of this study, do you have some advice for those who are just beginning their careers and are very interested in this? Where should they begin? They're going to contact you, but where should they begin? Where do they start?
Just contact me. We'll have a Zoom meeting. I'll talk you through it in half an hour and answer all your questions. It's not rocket science at all. Get yourself a partner in plastic surgery. Our plastic surgeon is pediatric, and we'll talk to them. And you know how to do biopsies. You know how to do wound assessments and surveys. You got this. This is not a big deal. You follow the instructions. If you don't have a study team or study coordinator, I'll send you resumes on how to hire people. This is not rocket science. And if it's people power, when the technology is ready and we got patients waiting, let's just do this together. It's not a big deal. And I'm glad to help you and hold your hand and be your partner in this.
Wonderful. Well, I can't thank both of you enough for being here today. This has been a wonderful webinar, so much amazing information and so much amazing work being done So thank you both very, very much for all your work.
Thank you. Thank you, Jean. Thank you, Jody. Thank you to our Pedra group.
All right, if anybody has any questions, you can follow up with Pedra at info@pedraresearch.org. We can help make you connected with Dr. Tang and Mr. Amoroso and Abiona. Thank you so much for joining us today, and we'll see you all again at the next webinar. Thank you.